Hi there, welcome to Mind Builders Hub. And for today, we're going to discuss the cell division and cell cycle. Cell division is a fundamental biological process that plays a pivotal role in the growth, development, maintenance, and reproduction of living organisms. It is a mechanism by which a single parent cell gives rise to two or more daughter cells, and cell division is a highly regulated and precisely orchestrated process, vital for maintaining the integrity of an organism cells and the transmission of genetic information. There are two primary types of cell division. Those are mitosis and meiosis, and each serving distinct functions. So, I'm June Sabate, and welcome to Cell Cycle and Cell Division. For our chapter outline, we're going to discuss number 1, the cell division, number 2, the importance of cell division, number 3, the prokaryotic and eukaryotic division, and last but not the least, it's the cell cycle. In here, let us check your knowledge first. Identify what is being asked for. It is the process by which the cell divides to form two new cells. Is it cell division, mitosis, binary fission, or meiosis? And the answer is cell division. Let's have another question here. The process that results in the division of a eukaryotic cell's nucleus into two genetically identical daughter nuclei. Is it cell division, mitosis, binary fission, or meiosis? The answer is mitosis. And third, specialized type cell division that occurs in sexually reproducing organisms. Is it cell division, binary fission, mitosis, or meiosis? And the answer is meiosis. Now, for our objective of today, we are going to define what cell division is. Next is to identify the purpose of cell division. Let's also discuss or describe cell division in prokaryotic and eukaryotic cell. And let us describe the cell cycle. So let's begin with the cell division. Cell division is a process by which a cell divides to form two new cells. There are three types actually. In the prokaryotes or bacteria, they have binary fission. And we have two for the eukaryotic, and those are mitosis and meiosis. So to begin with, let's talk about the prokaryotes. They are dividing, forming two new identical cells. In eukaryotes, like mitosis, the results in the division of eukaryotic cells nucleus has two identical daughter nuclei. That is for replacement or repair of damaged cells. Another thing is meiosis, and that is for or occurs in sexually reproducing organisms. It will lead to the formation of haploid gametes. Those are the sperm and the egg cells. We call them gametes. And there are a two-step process that reduces the chromosome number by half and generates genetically diverse offspring. So for the question, why do cells divide? Well, for some reason, cells divide for growth, for development, repair of worn-out tissues, and reproduction. Another is that to facilitate the exchange of materials, and of course, to control DNA overloading. So those are the answers why do cells divide. Now, in the continuation of our lecture, prokaryotic cell division will be the first. Again, they undergo binary fission. And in binary fission, there are three major steps that we need to determine. First, the DNA replication. DNA is copied, resulting into two identical chromosomes. This is how they copied and replicate chromosomes. Or to make it easier for our understanding, You will see in the second animation and how they replicate the DNA. Second, the chromosome segregation. In the chromosome segregation, chromosomes are separated and move towards ends to the poles of the cell, like this one. In cytokinesis, or separation, cytoplasm divides, forming two cells, like here in this animation and how they divide. So then again, they undergo DNA replication, followed by chromosome segregation, and during the separation, they produce genetically identical cells. And take note about this, each new daughter cell is genetically identical to the parent cell. And I want you to look at this animation showing on how they reproduce their cell. Next, let's move on to eukaryotic cell division. In a eukaryotic cell division, it composed of two main steps. Those are mitosis and cytokinesis. Each daughter cell is genetically identical to parent cell, like in binary fission. Cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent cell. And that is the first one, mitosis. In mitosis, there are four steps to follow. One is prophase, followed by metaphase, and third, anaphase, and last, the telophase. Cytoplasm divides, forming two new daughter cells after that. So we call it cytokinesis. So let's begin with identifying mitosis. Remember, in mitosis, it is a form of cell division that results in the production of two genetically identical daughter cells. And it is essential for the growth, repair, and maintenance of multicellular organisms. It always occurs in somatic cells, which make up the body of the organism. And the process ensures that each daughter cell receives a complete set of chromosomes and is identical to the parent cell. It is characterized by distinct phase, including the four that I mentioned, followed by cytokinesis, which divides the cell cytoplasm and organelles into two daughter cells. Now, in the prophase, you will see that the chromatin, which is a complex of DNA and proteins, condenses into visible chromosomes, and each chromosome consists of two identical sister chromatids connected by a centromere. Kindly take a look on the animation. So the chromatin, a complex DNA and proteins, condenses. Next, after that, you will see each chromosome consists of two identical cells or sister chromatids connected by a centromere. And this is a centromere. Next, the nuclear envelope begins to break down, allowing the mitotic spindle, a structure composed of microtubules, to form in the cell, like in this animation. Look at the mitotic spindle and the microtubules. Next, the spindle fibers attached to the centromeres of the chromosomes starts to move the chromosomes toward the cell's equatorial plane. Like in this animation, they are now moving. After the prophase begins with the metaphase. In the metaphase, the chromosomes align along the cell's equatorial plane, known as the metaphase plate. and kindly take a look at on how they align themselves in the cell equatorial plane. Next is that this alignment ensures that each daughter cell will receive an equal and complete set of chromosomes during cell division. Now, after the metaphase, let's move on to the anaphase. In the anaphase, it is characterized by the separation of the sister chromatids. After separation, the centromere of each chromosome splits, allowing the spindle fibers to pull the sister chromatids to opposite ends of the cell. And as a result, each daughter cells will receive a full set of chromosomes, like in this animation. Next, followed by the telophase. In the telophase, it marks the end of the nuclear division. And you will see that these are the key events that includes the separation. The separated chromatids arrived at the opposite poles of the cell. After that, the chromosome begins to decondense back into chromatin, like in this animation. After that, a new nuclear envelope forms around each set of chromosomes, resulting in the formation of two distinct nuclei. In cytokinesis, cytokinesis is the final step in the cell cycle and occurs simultaneously with telophase. It involves the division of the cell cytoplasm, organelles, and other cellular components into two daughter cells. And in cytokinesis, in animal cells, a contractile ring composed of actin filament pinches the cell membrane, creating two separate daughter cells. That happens in the animal cells, creating two daughter cells. Next, when it comes to the plant cells, a new cell wall forms down the middle, ultimately dividing the cell into two. Like here, you'll see that it's already forming and separating. So the end result of mitosis is the production of the two genetically identical daughter cells, each with the same number of chromosomes as the parent cell. This ensures that the daughter cells can carry out the same function as the parent cell and maintain the overall structure and function of the organism. Now, let's move on to meiosis. And it is a kind of cell division that results in 4 daughter cells. It composed of 2 main steps, the meiosis 1 and meiosis 2. In here, remember that meiosis is a specialized type of cell division that occurs in sexually reproducing animals or organisms, leading to the formation of a haploid gametes, the sperm and egg cells. And unlike mitosis, which results in 2 genetically identical daughter cells, meiosis is a 2-step process that reduces the chromosomes number by half and generates genetically diverse offspring. And here we go. Again, they are not genetically identical to the parent cell. Beginning with the interphase, then after that, the prophase 1. So in the prophase 1, you will see on how they move during several critical events like chromosomes condense and become visible, another homologous chromosomes, which are similar but not identical, pair up in a process called synapsis. And genetically material is exchanged between non-sister chromatids of homologous chromosomes through a process called crossing over. And this results in genetic recombination and introduces genetic diversity. And look at the nuclear envelope. It may break down and the spindle apparatus forms. In the metaphase 1, homologous chromosome pairs align along the cell equatorial plane. The orientation of each pair is random, contributing to genetic diversity. Kindly take a look on the chromatids. Next, in the anaphase 1, kindly look at this. The homologous chromosomes are pulled apart and moved to opposite poles of the cell. Unlike mitosis, where sister chromatids are separated, meiosis 1 separates homologous chromosomes. And lastly, in telophase 1, a new nuclear envelope forms around each set of chromosomes at the cell's poles. The cell then undergoes cytokinesis, dividing into two daughter cells. These cells are now haploid, as they have half the chromosomes number of the original cell. Continue. In the prophase 1, we are now moving on the meiosis 2, which is similar to mitosis, but it occurs in haploid cells and further reduces chromosome numbers. The two haploid daughter cells from meiosis 1 each undergo their own meiosis 2. So in the prophase 2, chromosomes condense and spindle apparatus forms in each of the haploid daughter cells. In metaphase 2, chromosomes line up along the equatorial plane in each haploid cell. In anaphase 2, the sister chromatids of each chromosomes are separated and pulled to opposite poles of the cell. And in telophase 2, look at this, is marked by the formation of new nuclear envelopes around the separated chromatids. Cytokinesis 2 follows, resulting in the formation of a total of 4 haploid daughter cells. So at the end result, the 4 haploid daughter cells produced by meiosis are genetically distinct due to the random assortment of homologous chromosomes in meiosis 1 and crossing over events, and these haploid cells are gametes, sperm and egg cells, in animals or spores in some plants and fungi. When two gametes, one formed from each parent, combine during fertilization, they restore the diploid chromosome number and create a genetically diverse zygote, which is the first cell of a new individual. Next is the cell cycle. In the cell cycle, it is a highly regulated process that describes the series of events during or occur in a eukaryotic cell's life. And the main point here is the arise and divide. It consists of periods of growth and development, DNA replication, preparation for division, and cell division. The two main periods include interphase and mitotic phase. In the interphase, it is the period of growth and DNA replication between cell divisions. Next, the 3 phases includes the G1 phase, the S phase, and the G2 phase. In the G1, look at the animation, the cell increased in size. After that is the S phase, or called the synthesis phase, wherein the replication of DNA happens and the 2 sister strands of DNA called chromatids are produced. Then lastly, the G2 phase. In the G2 phase, or growth phase 2, happens when the organelles doubled, a new cytoplasm forms, and all other structures needed for mitosis form. And that's it for the cell division and cell cycle. Thank you very much for watching, and I hope you learned something from our discussion today. And don't forget to subscribe the Mind Builders Hub videos. Thank you and bye-bye.